All right, hello everyone. Here is my little video about three-point crosses. So when I was in genetics, this was a big deal. It featured heavily on exams. Um, I'm not sure if that'll be the case, but this is important stuff. And so I thought we could practice it so that it's not some big crazy thing that is a hurdle for us on the exam, but instead that we can just like steamroll it and have it be something that we look forward to rather than dread. So 3-point crosses, unlike 2-point crosses, 3-point crosses are all about a cross at 3 different points, 3 different gene loci. And so we can start out right, let me grab the pen. Which color should I use? I'll use this one. We can start out right here at this parent, or I shouldn't say that. So yeah, this is actually our parent. And we see that there's a red chromosome and a blue chromosome. So this red chromosome has A alleles here at this locus, and this blue chromosome also has A alleles at this locus. And so because they have the same information, same homo information log, they are homologous to each other because they have on the same relative point on the chromosome, they have the same information. Right here, they have A, here they have the B genes, and here they have the C genes. So this red chromosome is homologous to this blue chromosome. This blue chromosome is homologous to this red chromosome. And obviously, this red chromosome has one chromatid and its sister chromatid. These two red chromatids are sister to each other. And just like these two blue chromatids are sister, actually, these two blue chromatids are sister to each other. So we have, and this is one individual, one parent, and we can obviously see that they are 2N because they have N and N, 2 chromosomes that provide information, genetic information at all the different loci, specifically A, B, and C. And so the genotype of this individual, this parent, is going to be, we can write it as A, B, B, C, C, because this chromatid and this chromatid, they contain the same information. They're sisters, they're identical. So we're going to have this capital A provided by one of the chromosomes, and this lowercase a provided by the other chromosome. Same for this B and that B, this C and this C. And another way to write this genotype that we see is A, B, C, A, B, C. And this can help us visualize what we see happening later, because when this parent goes through meiosis, something can happen that we know as crossing over. And I don't think I have to explain crossing over. We can see it here even, where we have 2 homologous chromosomes, the red and the blue, and they, at a certain locus, a certain place on the gene, or excuse me, on the chromosome, they can touch and then switch their information. So what happens between A and B, or yeah, here we have crossing over at a specific locus that is in between the A locus and the B locus. And what happens is that this red gene here will switch places. It'll come right here, and this blue gene will come right here. And that's what we see here. We are really only looking at these 2 chromatids because obviously it's only the chromatids that are closest to each other that are going to cross over. And so when we look down here at what's happened, this A, this red A comes, well, actually, I guess that's a misrepresentation. So what really happens, I guess they're trying to show, is that lowercase or blue B and blue C come up here to sit next to red A. And that's what we see, blue B, blue C next to red A. And then conversely, this red B and red C will jump down here to be with blue A. So blue A with red B, red C. And obviously the same can happen at not just the locus in between A and B, but the locus in between B and C. And something else that can happen is that not only do we have a crossing over between A and B, but also between B and C happening within or among the same two chromatids. So it crosses over at this point and at this point at the same time. And this can help us, this process, whether or not something happens at just one locus or at two loci, or even not at all, can help us to identify the placement of genes on a chromosome. And it's actually quite easy. And we have talked about it in lecture a little bit, but I wanted to kind of give the rationale behind it rather than just blatantly speaking about it. So crossing over doesn't always happen. If you think about you are, standing in a field and you're just hanging around, the likelihood that you don't get bitten by a random dog in the middle of this field is high, right? But it's possible that a random dog bites you. Jump. Pretend that's a dog. So the likelihood that a random dog comes up and bites you is less likely. So it's likely that you're happy. No one bites you. It's less likely that a dog comes up and bites you in the middle of a field. And it's even less likely that two random dogs come up and bite you in the middle of a field. So if we did this, if we stood in a field a bunch of times and we recorded what happened to us, the number of times, let's say out of 1,000, that nothing happened to us, that no dogs bit us, let's say that would be like 80% of the time. So 800 occurrences, no dogs bit us. And then less likely, let's say that 180 out of 1,000 times, one dog came up and bit us. And then even less likely, 20 of the 1,000 times, two dogs came up and bit us. And the same logic goes with crossing over, because chromatids don't always just like join up. Two chromatids, each one being from one homologous chromosome and one from the other, they don't always cross over. So that corresponds to this, no crossing over. A single crossing over event corresponds to this, where it's less likely than nothing happening, but still not as unlikely as a double crossover, getting bit twice. And so when we look at, or if we are given, well, I suppose if we look at the number of progeny that show recombination among, oops, that was not supposed to be the eraser, among the parental genotype, we can identify which one or which offspring, progeny that result are double crossovers, single crossovers, or even just the parentals, just by looking at their numbers. And so let's explore that right now. And actually, I think it would be more helpful to start with this one. So we're familiar with this one. We talked about it in class. But try to forget your preconceptions about this, and let's just do our best and talk about it. So like we just talked about, we have all of these Sorry, I'm trying to decide which color I want to use. Let's go with this one. So we have all these progeny, and we can see what number they occur in out of 755 total. Ooh, let me bring that up really fast. Anyway, yeah. So as we talked about with the dogs, it's very likely that you don't get bit at all when you're standing in a random field. And similarly, it's very likely that no crossing over happens in meiosis between one of the parents. Oh, and actually, well, we don't need the parent information. We can infer it based on our what we just talked about the dogs in the field. So it's very likely that no dogs bite you. It's very likely that crossing over doesn't happen. So the largest numbers of progeny that we see, like the overwhelming majority, are these two genotypes. And that means that because it's unlikely crossing over happens, it's very likely that we have offspring that show the same genotype as the parent. And we call this being non-recombinant. They are not recombining. But I like to call them parental, just so that we don't accidentally mix up recombinant and non-recombinant. It's also much easier to say. So these are our parental genotypes, our parental progeny. This 283 individuals and 278. And so we can look at their genotypes and see that because in this graphic, we weren't explicitly told what the parental genotypes are. We actually were in a previous slide. These are just the slides from our lecture. But if we weren't told, we could tell what the parental genotypes were just based on the large number of parental progeny. And so remembering, looking back at the previous slide, we had those two chromatids or chromosomes, and we had just these two chromatids crossing over. So we can kind of ignore the bottom chromatid. And, you know, they're all the same. They're STESS, STESS, SESS. So we can, based on our knowledge of crossing over and what we saw in that example, just examine the top, because this would be the only of these two chromatids that we see that would have experienced crossing over. So one of the parental genotypes was this ST plus E plus SS plus, and the other one was a simple STESS. So this was crossed with this. And what we want to do is determine what we really want to do is find the order of genes. We want to make a map. We want to know who is where and how far away they are from each other. So the way we do that is by observing the number of progeny that are exemplary of crossing over happening at the different loci between these two parents. And the best way that we can do that is to observe a non-recombinant progeny, you know, like these two parents that we see, or parental genotypes that we see, and offspring that exhibit crossing over at both of the loci. If you remember back to the dogs, it is very unlikely that you'd be bitten twice by two random dogs in the middle of a field, just like it is very unlikely, the least likely scenario, that double crossing or that two chromatids, chromosomes, cross over twice within a single crossing over event. And so the least likely progeny are going to be, or the double crossovers are going to be the least likely and therefore the lowest number of progeny. And so how can we use these two crossing over states, whether or not crossing over happened twice versus none, how can we use that to determine which gene is in the middle so that we can more accurately figure out our map and the distances? Well, it goes back to the last slide, and we see that we have STE, SS, ST plus, E plus, and SS plus. So what we can do is look at these double crossovers, look at their genotype, and compare them to one of the two non-recombinant or parental genotypes. So let's look at this one first. So we have ST plus E plus SS. And so the best way to compare them is to look or compare this to whichever of the two parental genotypes are the most similar to it. And in that case, we have ST plus E plus SS, just like we have ST plus E plus and SS plus. So only one of them is different, and it's SS. And from this, we know that it's SS that's in the middle. It is the middle gene. If we wanted to, we should visualize that just in case it's not intuitive. So what, actually, I'm also going to erase that. And so what we have is that because SS is in the middle, so let's rewrite it and let's take a look back at this parental genotype. So we had our ST plus SS plus, which we know is in the middle. Sorry, ant. Sorry, there's an ant on my desk and I had to blow it away. But so yeah, we had this ST plus, SS plus, and E plus. And this crossed over with our other parental genotype because, this individual parent crossed with this individual parent. So we had ST, SS, and E, because, ST, SS, E. And what happened is that at this locus in between ST plus, or in between the SS gene and the ST gene, there was crossing over that took place. So this ST came over here and became ST plus. Actually, that's not a good way to think about it. We had this ST that crossed over and is now connected with the SS and the E. And similarly, the crossing over happened between the other ones. And we had the ST going with the SS plus and the E plus. But also, because double crossover, the double happened, or excuse me, crossing over also happened at the other locus between the SS gene and the E gene. Crossing over happened. I'm going to use a different color that's more apparent. Oops. Okay, sorry. Let's just use an eraser. Dun, dun, dun. Sorry. I don't always know what I'm doing. Actually, I rarely know what I'm doing. So that shouldn't be the eraser. There we go. That's better. And so what happened is that because crossing over happened between two loci, this E plus went over here, this ST plus went over here. So these two are kind of like traveling in a pair, going to the other side. So it looks like it's the SS that's different, that the SS traveled, but in reality, what happened is that these two traveled over in a pair. And therefore, from this, we get that ST plus, SS, and E plus, because ST plus and E plus move down here. So we have our ST plus, SS, and E plus. And similarly, I'm running out of colors. We had our ST and our E traveling together, jumping over to this side to meet up with our SS plus. So we had ST and E jumping from the bottom to the top. So ST and E, and then we had our SS plus in the middle. So this is what's really happening with our double crossovers, but we can very easily take this shortcut and compare two similar, or a double crossover individual with one of the parental individuals that is most similar to it, and then see which one of them is different because of this thing that we saw. And that's the nice, easy shortcut to determining which gene is in the middle. So now we know, can I erase all this? So now we know that SS is our middle gene. SS is the gene that in reality, on the actual chromosome, is in between, I always forget to do that, is in between the SS gene locus, or ST gene locus, excuse me, and the E gene locus. So we can even rewrite it. That helps me. You don't, I recommend rewriting it. It just makes it more easy to follow, and you don't ever have to like do any rememberings or conversions of anything. Just nice and clean. So this is what our parents really looked like. And so we also have, where do I want to go with that? So we found it and we've rearranged our parents. And we know that our gene map is going to look something like this. SS in the middle and ST either on this side, or it could be on this side, you know, we don't really know. It could be this way, or it could be this way. We don't really know, and it doesn't really matter right now because we don't have other genes on the other side to anchor ST either over on this side or on this side. So just for now, knowing that SS is in the middle is what's important, and I should not have drawn that right where I need to write the rest of my stuff. Dun, dun, dun. Okay, and we also want to change the color. So this is what our map is going to look like. It's going to be something along the lines of SS, E, that is E, ST. And so now we need to find the distances, the relative distances, the map distances between the ST locus on our chromosome and the SS locus on our chromosome. We also need to find the difference, or the distance between the SS locus on our chromosome and the E locus. So we can determine this distance and this distance. Oops. Okay. So how do we do that? First, let's focus on finding the distance. What color should I use? The distance between ST and SS. So the distance between ST and SS, we have this crossing over event, this single crossover. So let's look at the first single crossover, or single crossover 1. Single crossover 1 is going to occur between, oh, yeah, I should have rewritten that. I knew I was forgetting something. So let's rewrite, or let's redraw our cross. Sorry for not being better about this. So we have our guy. Oh, that's what I did wrong. Okay, never mind. So we have our first parent that was ST, SS, E. And we have our second parent who is ST plus, SS plus, and E plus. Oh, I also forgot the line. Goodness gracious. Bing and bong. Okay. So we have a single crossover at this locus, which means we're going to have this and this. So single crossover 1 occurs at the locus between the ST gene and the SS gene. And the genotypes that come out of it, are going to be we can just follow this line. So we have ST combined with SS plus E plus. So ST combined with SS plus E plus is one of them. And then the other one is ST plus with SS and E. So the other genotype is ST plus SSE. These are our two genotypes, or two recombinants that result from the single crossover and only the single crossover. And we can, look back. I'm going to do that because it's bothering me. And we can look back at our original progeny and determine which genotypes correspond to these two genotypes. So we can see that, look at the numbers. So ST plus with an SS, or no, excuse me, an ST with an SS plus and an E plus. And we see that right here, ST with an SS plus and an E plus, ST with an SS plus and an E plus. Very good. So that number is our 52. And then an ST plus with two non-plussed, I don't know, a better way to say that, ST plus with SS and E. ST plus with SS and E are 50. Sorry, my handwriting's not the best. So there are 52 and 50 of these individuals that saw a crossing over happen only at the locus between the ST and SS genes. And so now that we have this, we can find the distance between the ST and SS genes. But something that we have to consider is not only these two, but also our double crossovers. Because in order for them to have been a double crossover in the first place, they had crossing over at both the first and second loci. So they did have this event happen to them where ST crossed over, or where the two chromatids crossed over at this locus, at this first locus. And so they do contribute to the distance between ST and SS. So not only do we add 52 and 50, we also add the 3 and the 5 are two double crossovers, because in order for them to have been a double crossover, they also had to have had both of the single crossovers. So those were 5 and 3. And so in order to find the frequency of individuals, we just add the number of individuals that saw this event happen, a crossing over event at the first locus. So the number of individuals who saw that, which is 52 plus 50, I'm actually going to switch back to blue. So 52 plus 50 plus 3 plus 5, or 5 plus 3. So all of these individuals had crossing over at this first locus, at the very least. And then we just divide that by the total number of individuals. And what's the math on that? 14.6%. And so now we can investigate. Oh, that should be the eraser. The one time I need it to be the eraser, it's not. So we also have single crossover 2, or crossing over here at this second locus between the SS gene and the E gene. And so just like we did before, we are going to see crossing over happen right here and right here at this locus. And so we can once again follow the line ESS plus ST plus, or actually better not to do it backward, ST plus SS plus E. So ST plus SS plus E. And also, oh no, dang it, I forgot about that. Shoot, sorry. But we can do it. We can make up for it. Good job. So, and then also we have ST, SS, E plus. These are our offspring, our progeny that come from this single crossover event. And so we can look for this genotype, ST plus, SS plus, E plus. We look for them over here. ST plus, SS plus, E plus, perfect. So that's 43. And then obviously we've narrowed it down, but we can just, for the sake of being thorough, look for ST, SS, and E plus, ST, SS, E plus, perfect. And that's 41. I don't draw my fours like that. What's happening? 41. And once again, we can find now the distance between SS and E, but don't forget the double crossovers also had crossing over at this second locus. So we do have to consider them when finding the distance between SS and E. So we have our 43 plus 41 plus 5 plus 3. And I'm also going to clean this up with my colors so that it's nice and pretty so that we can keep everything straight. Divided by our total. So the number of individuals who showed or experienced crossing over at the second locus divided by the total number of individuals gives us the frequency of individuals that had crossing over at the second locus. And this is 12.2%. And I'm also, I said I was going to fix it, but I hadn't yet. Dun, dun, dun. I'm going to rewrite that because it's not very visible. So this was our 12.2%. We divide by 755 to get 14.6%. And so, as our final answer, we have discovered that SS is the gene in the middle. It is in between the genes at the ST and E loci. The distance between ST and SS is this single crossover. So 14.6 map units, or 14.6% recombination. And then the second crossover is 12.2%. So this guy is a little bit closer than this guy. And that is a triple, or a 3-point cross, excuse me. Great job, everyone. We did it. And so with that under our belt, I think we should explore another one. Let's clear all this. Jump to the next one. I'm glad I did this one first. This one would have been a nightmare otherwise. And let me pull that up just really fast. Sorry. Please hurry. Just kidding. We don't have to hurry. We're patient. I just need the math. Okay, so we have another one. Perfect. No, stop. Video. Leave me alone. So we have another one. Let's bring out the pen. Okay, so two Drosophila flies are crossed. Oh, yeah, we're going to be dealing a lot with Drosophila. So, but this one is going to be easier because we have a little trick that we used with the genotypes that we see. So two Drosophila flies are crossed in a typical 3-point cross and yield the following phenotypes among the progeny. So it's pretty much the same that we had last time, except just different numbers. And we're no longer dealing with SS's, STs, and Es. We're dealing with A's, B's, and C's. Give the parental genotypes and the map distances. So the same thing that we did last time. So where do we start? We start with identifying the parental, or the individuals that show the parental genotype. Just like standing in a field, not being bitten by dogs, the parentals are the most likely because it is the most likely that, or the parentals are the most numerous because it is the most likely that crossing over doesn't happen. So we have one parental here and another parental here. Similarly, being bitten by a dog twice is very unlikely, the least likely, and therefore the least numerous on the number of occasions. So I'm also going to switch colors. So we have our double crossovers here. And so we can compare one of our double crossovers to the parental genotype that's the most similar to it. And in reality, this is like A plus, B plus, C plus, but it's even easier to visualize here because we can just look at which one is different based on whether or not it shows us a letter or a plus. So we can compare, or actually, it's always helpful to rewrite the parental genotypes. So we have one that is obviously plus, plus, plus. Then we have another, it is crossed with A, B, C. And so how can we determine which one is in the middle? We can compare this one, this double crossover, to the parental genotypes that's the most similar to it. And in this case, it's easy to tell because we have BC, BC, and then one of the pluses is different. So obviously it's different here because B is similar here, C is similar here, but we have this A locus that's different. So we know that A is the one in the center. It is in between B and C. And just to compare and make sure we're doing a good job, this one is similar to this one. And this one is similar to this one. So the B locus is also B plus in both the parental and double crossover. The C locus is the same as, the C locus in the double crossover is the same as in the parental, and it's once again the A that's different. For me, it's easier to visualize this pluses than writing out all this stuff. But if that doesn't help you, feel free to write that stuff. Anyway, so we have identified that it is the A locus, or the A gene that's in the middle, so we can even rewrite this to be B, A, C. A is in the middle, and so we can rewrite our guys, and we know that the cross is in between plus, 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 and actually it's going to make the formatting a lot more palatable if we did that first. Plus, plus, plus. So this is the cross that we're dealing with, the original cross between 2 parents. And so we have identified the order of our genes. The overall thing that we're looking for is going to be this map, which gives us B, A, and C. I never write letters the same way twice, and it's frustrating because sometimes I write them really ugly. Grr. We can do it. Good enough. B, A, C. Actually, it's not good enough. Sorry. Yeah, that's good enough. And so now we need to find distances between the genes at the different loci, specifically at this one and at this one. Actually, I'm even going to rewrite that. Between, which color should we use? We did blue and lavender last time, so let's do, I hate orange, let's do maroon. and I really don't have a lot of colors to work with. Maroon and pink, why don't we? And so this is going to be our single crossover 1. This one, our single crossover 2. And now that we know who is the non-recombinant, the parental, who are the double crossovers, actually, I should not be using the single crossover 2 color. I should be using a distinct color. We have been using this nice pale green double crossover. Okay, so we've identified the middle gene. It's very good. And now we can find the single crossovers. So let's look for single crossover 1 first. So single crossover 1 is going to happen between the B and A genes. Locus in between the B and A genes. So right here. So we would see this happening. So we can follow it. One of the genotypes that we see is going to be plus AC, so plus AC, and the other one is going to be B plus plus. B plus plus. We can look for these genotypes among our individuals, plus AC, and then B plus plus. So we have 81 and 75, specifically the A plus C, or the plus AC is 81, and the B plus plus is 75. So we can combine these. We have our, oh my God. We have our 81 plus 75 plus, I'm going to make the double crossover color yellow for some reason, 3 plus 1, hopefully that's not too difficult to see, and then divide it by the total number of progeny. So the total number of progeny, or the frequency, excuse me, of the progeny that showed this single crossover 1 is the number of individuals that showed this single crossover event divided by the total number of individuals. And we have the math right here. This equals 0.08. So this distance is 0. Or actually that's just 8%. One day I will learn how to write the number 8 without having to rewrite it 6 times. So now we have done single crossover 1. Let us now find out single crossover 2. So single crossover 2 happens right here between A and C. And so we would have BA crossing over, BA crossing over. And so we would have, if we followed it, plus plus C as one of the genotypes. And then BA plus. We'll follow that in a more followable way. BA plus. BA plus. Plus plus C is this genotype. So 90 individuals. BA plus is this one. So 86 individuals. Once again, frequency of individuals, or the frequency, excuse me, of individuals that had crossover 2 or single crossover 2 is the frequency or the number of individuals who showed that crossing over divided by the total number. One day I will be able to say that without stumbling. Divided by the total number of individuals. This is equal to 0.09, which is 9%. See how easy that was? We just needed to lay down the foundation, and we have our answer right here. We have the order, the distances between each of the individuals. We are doing fine. 3-point crosses, easy mode. I have another one for you. Oh, do I have one that's in trans? That would be a really good example. Let me see really fast. Ooh, is that this one? Oh my God, I think that's this one. That would be so cool. But Blackboard always messes with me. Why does Blackboard do this? Blackboard, when you do this? Okay, that is not the right one. Let me find it really fast. Sorry. I can clear this first though, and then go to it. I don't think I've ever watched this one. Is this one in trans? I think this one's in trans. Okay, let's do it. I believe in us. Wait a minute. This isn't the same one. Sorry, 1 second. Oh no, it's not in trans. That's boring. It's okay. It would still help us to do it. It's right here. No more Drosophila this time. This time we're dealing with plants, and plants are cool. We like plants. Sorry, just one moment. Oh my God, what happened to it? Hey. You know you want to. Come on. Okay, there we go. Sorry about that. Okay, so moving on. You guys can do this in your sleep now. So very quickly, let's use dark blue. So it's most likely that we don't get bitten by a dog. It's most likely that we don't have crossing over. So those are the most frequent numbers. It's least likely that we get bitten twice. Those are the least frequent numbers. So we have our double crossovers and our parentals. Oh yeah, I can read the question though. Plants with genotype X plus Y plus Z plus are crossed with plants having the genotype XYZ. A 3-point cross is performed using the F1 progeny and a plant with the genotype XYZ. Below are the progeny produced. Use the data to determine the order of the genes and the distance between them on the chromosome. So, once again, easy mode, we can do this. First step is to, very good, find the middle gene. And I'm just going to do pluses, not X plus, Y plus, Z plus. Just going to write them as pluses because it's faster and it helps me visualize it better. If it doesn't help you visualize it better, then feel free to write X plus, Y plus, Z plus. So we have our parentals, which are going to give us, we have an X, Y, Z crossed with a, actually, crossed. Sorry, I just realized it would be confusing to write this as crossed because we also have X as an allele. So we have XYZ crossed with plus, plus, plus. And this, we see it as, actually, no, I shouldn't rewrite it yet. XYZ crossed with plus, plus, plus. These are our 2 parental genotypes. Then we have our double crossovers. Let's compare this one. So we have plus Y, Z. Which one is the most similar? Obviously, this one. So it's the X that's different because we have the Y and the Z that are the same. And remember, that's in reality because we have Y, X, and Z. Or actually, excuse me. We have Y, X, and Z. And these two are kind of moving together, switching over, such that we end up with X or Y. Goodness. And then the plus, that represents our X plus. And then Z. So this is the double crossover. And we know that X is the one in the center. So we know that X is our central gene. So we can rewrite the parentals as being Y, X, Z. Ooh, actually, I always forget to do this. Gotta be consistent with your colors, you know? Y, X, Z as the parentals crossed with plus, plus, plus. And so we can investigate our, our single crossover 1, which happens between the X and the Y, or rather between the Y and the X. This is our single crossover 1. And then we have our single crossover 2. So let's investigate single crossover. You know, let's be cheeky and investigate single crossover 2 first. So single crossover 2 occurs here at this locus between the X and the Z genes. So we have this and this. So we can follow it along. We have YX plus as one of our genotypes, one of our possible recombinant genotypes. And we have plus plus Z. So YX plus is 39, and then plus plus Z plus plus Z, 41. So 39 here, 41 here. And we remember 39 plus 41 plus what? Oops, plus what? Exactly. I'm glad you didn't forget. I'm proud of you. All divided by the total, which is 732. And let's bring up the math really fast. That is going to be equal to 0.135. And let us now move on to single crossover 1, which happens right here. So we would have our crossing over. So one of the possible genotypes of our recombinant offspring, or excuse me, our recombinant offspring is going to be Y plus plus. Y plus plus, and the other is plus XZ. Plus XZ. So Y plus plus is Y plus plus 70. And we're kind of only left with this one, which we see is plus XZ plus XZ. Perfect. Always get to check, make sure you're doing it right. So we have 70 plus 66 divided by 730. I'm glad you caught me. Good job. Hee hee hee. Good job. And that is going to be equal to 0.212. So we can make our map. So which gene is in the middle? X. And then it could be Z and Y, or it could be Y and Z. And so we know that the distance, the relative distance between the Y and X genes are our first, or the recombination frequency at the locus between them, single crossover 1. So we have 21.2%. Actually, I should verify whether or not you answer in percent or in a decimal. Usually, I've noticed on all of Dr. Chiari's exams, at least, that you're asked for decimals, never do fractions. So let's be what we think is most accurate according to Dr. Chiari. Say that the distance between them is 0.212. And I'm also going to rewrite that in the proper. Actually, I don't need to rewrite it. And then the distance relatively between x and z is the recombination frequency between x and z, which is this single crossover 2, which is 0.135. We did it. This is super easy, right? You just need to be patient. Always remember your stuff. Parental's most frequent. Double crossovers least frequent. Try to write it this way is the way that I do it. That's what helps me. If you find a better way, do it that way. We didn't talk about 3-point crosses from trans chromosomes in class. So I just bet on not having to do trans 3-point crosses. What we've been doing are cis 3-point crosses, where let me back up even to that. So this is in cis configuration because, let me bring out the pen and make it a color that we can see, because we have capital A with capital B with capital C, with capital A with capital B with capital C. So what we see is that this chromatid crosses over with this chromatid, and this chromatid is entirely dominant. It only has one kind of allele to give, and this one also only has one kind of allele to give. It would be in trans if we had, for example, A, B, C, or even A, B, C, or even A, B, C. Because cis configuration is when you have all of the same kind of allele on one of the chromosomes that crosses over. So this is in cis because it's all capital letters crossing over. It would be in trans if we had some funky mixing, where then, just for example, we had this chromatid, was A, lowercase b, capital C, and then this one was lowercase a, capital B, lowercase c, because then when we cross over, the double crossover is going to be this B allele. And so the double crossovers are going to be ABC, all capitals, ABC, all lowercases. And these would be the least frequent individuals. As you know, likelihood of getting bitten by a dog twice. These would be the double crossovers in trans with these two parental genotypes. So honestly, it's not too much more complicated. You just have to pay a bit more attention, and it's truly no problem. We have the process. We know what we're doing. We could kill a trans cross either way, a trans 3-point cross. So yeah, how long is this? Oh, it's 1:43. I've missed my class. Well, I haven't missed it. It only started 13 minutes ago. Anyway, so if you have any questions, want me to elaborate on this at all, let me know. The quiz is all about 3-point crosses. You might even, actually, I'm not going to comment on that. But yeah, if you have any questions, ask Sam or I. And then if we don't answer, ask Dr. Chiari. And yeah, thanks for stopping by. Hopefully this wasn't too long. Let's cut the recording. Thank you again for watching.